All right, guys. So this is going to be the first day of our blood unit. This does have a little bit more vocabulary in it than some of our other units have had. So we are going to split this into two vocab quizzes. So you can see the first set of words is about 19, and I believe the second set of words is 19 or 20. So you will see two different vocab quizzes on the calendar as we are going through this. So whenever blood is found at scenes, it's often going to be violent crimes. So it may have been where someone was jumped. There may have been weapons involved, like a stabbing or a shooting, things like that. And investigators are going to be able to use the location of where they're finding blood, the distribution of blood. So is it all in one like pool, or is it spread out throughout the scene? Is it just on the floor, or is it also on the walls and the ceiling? And they can also look at the pattern of blood and blood stains to help them reconstruct the crime and what occurred there. Blood's general biological characteristics are considered class evidence. Whenever there's a transfer of blood between the victim, the suspect, and the crime scene. And the blood, including blood type, leads to a group of individuals, which is referring to that class evidence. So just finding blood patterns or being able to tell blood type is a class characteristic. You actually have to look at the DNA pattern itself whenever you're trying to individualize blood. Blood can be individualized whenever you're comparing or looking at DNA. And when what we believe is blood is found, it has to be tested to make sure it's blood. It might be some form of simulated blood. It might be mixed with something so it doesn't appear as if it's really blood, but it has to be tested to see if it actually is blood. And several presumptive tests are going to be run on the sample to confirm the presence of blood. And a presumptive test we mentioned last unit with the spot test. But a presumptive test shows that blood may be present. So if it changes color or reacts, it's likely that blood is present. But there can be false positives. There are some substances that will test positive for blood, even if blood is not present. False positives could come from stuff like bleach, plastic residues, potatoes, and even certain metals like iron, copper, cobalt, because those are also typically found in blood. So just because it tests positive for blood doesn't always mean that it is blood. There was a case, I want to say about 20 years ago, where they thought there was a previous pool of blood in the kitchen, and it turns out that's the corner where the potatoes are stored. And so it was testing positive for blood, even if they were not present. Many chemical tests will react with the hemoglobin in the blood. So whenever we have chemicals testing for blood, it is trying to search for that hemoglobin, and that hemoglobin is what it will react to. Hemoglobin is our oxygen carrier in our blood. It's what gives red blood cells their color. And yet again, these presumptive tests are still, or the chemical tests are still considered presumptive tests. So if there's a reaction, we still have to run further tests. Blood stains, so dried blood, can also be tested with luminol. And we've mentioned luminol whenever we did impressions, but luminol is a very sensitive chemical indicator for dried or washed blood. So if you remember indicators from chemistry with acids and bases, whenever an indicator came in contact with an acid or base, it changed color. This will do the same kind of thing, but luminol instead causes chemoluminescence. And chemoluminescence is the emission of light or glowing produced from a chemical reaction. So they will take luminol and spray where they believe blood is, and if it glows, that is testing positive for blood. You may have heard of similar reactions of like organisms that can glow, like lightning bugs, certain algae, things like that. That is bioluminescence because it's within a living being. But it's that same thing where there's a reaction causing light and that glowing. Once we find what we believe to be blood, we have to figure out if it's human blood or animal blood. So investigators have to determine what species the blood is from by using a Preston test. And Preston tests will distinguish between human and animal blood. So a normal Preston test contains an animal serum that contains antibodies specific to human antigens. And serum is a liquid that separates from clotted blood. So if you ever have a wound and it is kind of scabbing over, it can be a little oozy. That is the serum that is being excreted. Antibodies are proteins in our blood or other secretory fluids, so it can be in sweat and things like that tag, destroy, or neutralize bacteria, viruses, or other harmful toxins, producing an immune response. So what that means is we said it's an animal serum that has antibodies for human antigens. So whenever it comes in contact with human blood, there is a reaction that we see. Whereas if it is animal blood, we will not see a reaction. Whenever it said it reacts with human antigens, antigens are any foreign substance in the body that's capable of causing disease. And typically what happens is if something is in our body that is not supposed to be there, it triggers an immune response, and the body will produce antibodies to try to get rid of it. So if you get the flu, your body triggers and tries to produce antibodies to fight off the flu. It tries to attack that virus and whatever is not in your body, or what should not be in your body. The serum reacts with the antibodies to form agglutination whenever it comes in contact with human blood. And agglutination is an allergic reaction where red blood cells will clump together in response to certain antibodies. So anything that is not supposed to be in the body, the antibodies try to react and surround it for it to be filtered out of your body. But this can also happen if someone is not given the correct blood type. It can cause that immune response, and it can cause clotting and things like that. The Preston tests are so sensitive that even blood that's been diluted or dried or years old can still test positive. So even if we have a dried blood stain, or there's blood and someone poured bleach on it, or it's in water or something like that, we can still test positive to see if it is blood or if it is not. In these tests, anti-serums can also be used. Anti-serums can be human or animal serum that is specific to a certain antigen. So we'll typically see this whenever we are testing for blood types. We will have serums that specifically target the different antigens of the different blood types. Whenever we're looking at bodily fluids, serology is the study of bodily fluids. And this is because it not only looks at blood, but it can look at sweat, urine, things like that. But blood is a big part of this because about half the human body is blood. The fluid part of blood, which is your plasma, which we mentioned plasma last unit of that's where we can test for drugs. The plasma makes up about 55% of your blood. It's about 90% water, And then 10% are going to be metabolites, waste, salts, ions, and proteins. So things floating around within that plasma. Metabolites are the molecules involved during metabolism, so allowing us to break down materials. And the most common ions that are going to be found in your blood are sodium, chloride, and hydrogen carbonate, which is going to be a byproduct of your respiration, of your breathing. In your solid part of your blood, which is going to be floating around in the plasma, there are 3 different things that we have in there. We have our erythrocytes, which are red blood cells, which contain our hemoglobin. So that's what reacts with those Preston tests. Hemoglobin transports oxygen from the lungs to other cells of the body and then carries carbon dioxide back to the lungs to be exhaled. So the red blood cells are kind of like the cars that transport everything through our blood system. We also have our leukocytes, which are white blood cells. These are the primary cells of our immune system that produce antibodies. So the antibodies are actually produced by your white blood cells in your blood, essentially. And then your platelets are also floating around in there. And platelets help with clotting by initiating the formation of fibrin. And that clotted material from the platelets is what produces the serums that can be used in the Preston test. In 1901, Austrian biologist Karl Lansinger recognized that all human blood was not the same. They would have surgeries or give blood transfusions, and people would have these horrible reactions, throw blood clots, and die. And they couldn't figure out why this was occurring, why all blood was not the same. This led him to develop the ABO classification system after many blood transfusions resulted in immediate death for no apparent reason. And Lansinger also later developed the rhesus factor, or Rh factor, in blood. This is another antigen that can be tested for. This is in your blood types. If you're positive or negative, that is looking at that rhesus factor. And this further led to the development of over 100 different factors in blood. So there are individuals that have certain antigens or certain proteins that they produce. And so even they can test more than just blood type. So there are individuals that are they might have leukemia, that in order to help them, they need certain antigens. So there's only so many people that produce those particular antigens. So they can test for those things and those specific factors in blood. And in terms of DNA, theoretically, no 2 people, except for identical twins, have identical blood factors. And blood factors are just a combination of antigens, enzymes, and proteins in the blood. And like I said, someone that has leukemia or something, maybe they are deficient in certain proteins. So when they're analyzing donor blood, they might find blood that is high in certain proteins that they need. The blood factors are often looked at for blood typing as well. And whenever we find blood at a crime scene, it may not be appropriate to scan its DNA unless we have something to compare it to. But one of the quick and easy things we can do is look at blood typing, because individual blood analysis is time consuming and expensive. So if we're able to narrow down our suspect field based off of blood type, it's going to save us time and money throughout the investigation. The ABO and Rh blood characterization is so important because it can be done not only on whole blood, but also dried blood. So blood type is going to be one of the things that we can test even if the blood is old or contaminated. And also about 80% of the population are what we refer to as secretors. So these are people whose antigens are also found in other bodily fluids other than their blood. So they might be able to determine blood type from sweat, saliva, urine, earwax, snot, anything that comes out of the body. They can test for those antigens as well. So when looking at our blood cells, the antigens on blood cells are what is going to be responsible for your blood type. And then our white blood cells manufacture our protein antibodies. So our immune system all starts with our white blood cells. And the antibodies are meant to attack any invaders into your bloodstream. This might be any foreign antigens that are not supposed to be there. Venom, if you get bit by a snake or something, bacteria, someone else's blood, viruses. All of your immunity starts with your white blood cells that produce those antibodies. And when we're looking at blood type, like we said, your blood type is determined by your antigens. So if someone is type A blood, and I'm going to draw this out as simple as I can. If someone is type A blood, so they're red blood cell, I'm just going to write red cell, this would have A antigens around it. So these would actually stick out off of the blood cell essentially. And then our white blood cells would produce B antibodies that would code for different antigens. If we had type B blood, our red blood cell would have B antigens. And then our white blood cells would produce A antibodies. So your blood type is based off of what antigens are on your blood cells. So if we look at type AB blood, that means that they have both A and B antigens, and they don't have any blood type antibodies that they produce. Whereas if you're type O blood, think of O as in 0. So your red blood cell has no antigens on it. It is plain. And your white blood cells are going to produce antibodies for type B antigens and antibodies for type A antigens. But you can remember that there's only A and B antigens and antibodies. Whatever antigens you have match your blood type, and then you always produce the opposite antibodies because those are things that should not be in your system. How they kind of use this knowledge of the ABO classification system is whenever they're dealing with blood transfusions, blood interactions, and things like that. With antibodies, they do tend to be bivalent. So I did a very simplified drawing. But if you think of the bi means 2. So that means that antibodies have 2 sites that they can attach to antigens. So I should have drawn it a little bit more where there were 2 different sites for them to connect. So they're able to attach to 2 antigen sites, and what that does is whenever it starts linking these different blood cells together, it causes that agglutination and clotting or clumping. And this is where people were dying from blood transfusions due to this allergic reaction. So for example, if a type B person, I'm going to draw this at the bottom. So our B blood cells had triangles, and then it had the A antibodies, which were like the squares. If they're given type A blood, whenever someone receives blood, they're only receiving the red cells. They're not receiving their antibodies and stuff like that. But if someone receives type A blood, the little antigen on the type A blood will hook up with the antibody in the type B blood. And wherever this happens and every antigen gets an antibody attached to it, it starts causing these clots because you're having all of these cells and antibodies clumped together because it is something that should not be in that system. And this can be fatal if it is not caught immediately and these clots form. You're not circulating as much blood if it actually like blocks an artery and stuff. These are the types of things that can tend to cause strokes and stuff like that because it causes a blood clot. It is not circulating to the brain, not providing oxygen to the brain. The individual does have a stroke a lot of times in cases like this. This is why blood typing is a must before any type of blood transfusion, because if someone is given the wrong type of blood, it can be a pretty immediate reaction and it can spread very quickly. Our blood is pumping through our body at a pretty fast rate. And this not only has to be done with blood, but it also has to be done with plasma and whole blood transfusions. They have to make sure that the blood types match up and it's a safe interaction to prevent an immune response. So some people might get plasma transfusions or blood transfusions, depending on maybe they're anemic and things like that. Maybe they have a surgery and they need blood transfusions because they lose too much blood throughout the surgery. All of that has to be taken into account. So looking at safe blood factors that can mix together. And this is something very useful, especially if you have health issues or health concerns, or maybe you have donated blood, to know who your blood can go to. If you have type A blood, we said they have A antigens and B antibodies. So that means that they can donate blood to other people that also have A antigens. So that's going to be type A and type AB. They can only receive blood that does not have B antigens to react with their B antibodies. They can only receive blood from other people that are type A and people that are type O. If someone is type B, that means they have B antigens and A antibodies. So they can only donate to other people that also have B antigens. So that's going to be type B and type AB. And they can only receive blood from people that do not have A antigens to interact with their A antibodies. So that's going to be type B and type O. If someone is AB, like we said, that means that they have both A and B antigens and no antibodies. Since they have both antigens, they can only donate to people who also have both antigens. So type AB blood can only be given to someone else that is type AB blood. However, AB blood, because it has no antibodies, is referred to as the universal recipient. They can receive blood from anybody because their antibodies are not going to react with other blood types because they don't have them. And then if you're type O, you guys may have heard of type O is the universal donor. Since type O blood does not have any antigens, it cannot react with any antibodies from other people. So they can donate to anybody, any blood type. However, because they have both A and B antibodies, they cannot receive any blood that has any antigens. So they can only receive from other people that are type O. So make sure to look at this, and you're looking to see that whenever they're receiving blood, is it going to react with their antibodies? So we mentioned that Lansinger also developed the Rh factor, which was just another blood factor, another antigen. The Rh factor is important for serology, and this a lot of times is referred to as the D antigen. So yet again, it's a protein that is on the blood cell itself. People that have this antigen are said to be Rh positive. And this is where if someone says they're like O positive, that means they have type O blood and they are positive for this Rh antigen. There are about 85% of the population that are positive for the Rh factor. And then people without the antigen are referred to as Rh negative. So that might be if they're O negative, they're type O, and they do not have that antigen for the Rh factor. And this distribution can be different throughout the country. So there are some areas where you will have clusters of similar blood types. And that's something that they will kind of see. They don't really have a specific trend for, individuals with certain backgrounds tend to be positive versus negative. This can kind of go back to the Punnett squares from biology, looking at blood types, because they're looking at what factors do they get from the mother and what factors do they get from the father. Whenever we're looking at how common blood types are, this chart has the breakdown of the different percentages. So about 40% of people are type A blood, about 11% of people are type B, only 4% of the population is type AB, and then 45% of the population is type O. So if you've ever given blood, I'm O negative, so I get calls from the Red Cross all the time to donate blood. Even if I'm not eligible at the time, they're calling me because my blood can be given to anyone. But even though they are in high demand, it doesn't mean it's rare. But it's because it can be given to anyone that lessens the effects of that immune response. Whenever we're looking at the Rh factor, like we said, about 85% of the population are positive for that Rh antigen, 15% are negative. So if you're looking at this and kind of combining these percentages, and we're going to look at that similar to like we did probability in Unit 2, the most common blood type is O positive because those are the two that have the highest percentage. Our rarest blood type, the two that have the lowest percentages, are going to be AB negative. So if you happen to know your blood type, you can kind of figure out how common it is. So whenever we are blood typing, and a lot of times blood typing is used to exclude individuals because, like I said, we tend to do this at a crime scene and we'll test it with blood types or medical background from our suspects. But blood typing can typically show how two blood samples have different origins. So this is typically used to exclude suspects from our suspect pool. Because if we are narrowed down by blood type to one suspect, we can see what other evidence points towards that one suspect. If it leads us to two suspects, then we can run DNA that's more time consuming and more expensive to narrow it down between the two. And the distribution of blood types in a population can be used to statistically calculate the probability of a suspect having a certain blood type. So if we only have blood type and not DNA to go on, like if it's too degraded of a sample, whenever we take that evidence into court, even though it's class evidence, we would be able to support it based off of the rareness of whatever the blood type is. And that could help support the case. So when we're looking at probability, this we did back in Unit 2, I believe, where when we were looking at class evidence and we were looking at, there might be 1,000 people that have white shirts and 1,000 people that have blue jeans, but whenever we look at the amount of people that have both, it's only 200 people. So if we're looking at what part of the population would be B positive, we would have to multiply the two percentages together. So we said 11% of people were type B blood. There were 85% of people that were positive for that Rh factor. So if we multiply that together, that is 0.09. So if I make that a percentage, that would end up being 9.35% of people that would be B positive whenever I'm looking at that together. So like I said, the rarest blood type is that AB negative. If we're looking at the data for that one, we said only 4% of people were AB. 15% of people are negative. So if we multiply those two together, it's 0.006. So move the decimal over twice. That is 0.6% of the population that is AB negative. So if we happen to find AB negative at a crime scene, our suspect pool, there's probably not likely to be more than one person that is AB negative. So yet again, that can exclude a bunch of people from our suspect pool. And we're going to do blood testing one day after we get back, whenever that is. But blood can be tested for blood type by mixing a drop of blood with a drop of anti-serum that contains known antibodies. And if agglutination occurs, if we see clumping, we can use that to determine the blood type. Because if there is clumping, it means that that antigen is present in that blood. And whenever we're testing dried blood cells, the red blood cells have ruptured whenever they dry out, but the antigens are still there. So that's why it can still test, even if it's a dried, degraded, broken down sample. And one thing to make note of that we talked about in Unit 2 as well with evidence, if you have blood evidence, so you have like blood on a t-shirt or something like that, do not seal them in airtight containers. So you don't want to put them in a plastic bag, things like that, because they can mold and mildew. And if the sample gets damaged, it can no longer be used in court. That is where we are going to stop today. And we will pick up with whatever the next day comes. If we are also out tomorrow, the video will be looking at blood spatter and angle of impact, which is math-based, which we will also have to do something with after we get back.